Once again, I welcome you to BTN 311 class. In today's class, we shall be looking at respiration. Respiration is simply put as the opposite of photosynthesis. It is a series of cellular activities that involve the enzymatic breakdown of organic substances like carbohydrates, like your protein, like your fatty acid into carbon dioxide, water, and the release of energy in the process. The energy locked up in macromolecules, which are products of photosynthesis, are usually unlocked in respiration. Every macromolecule has to be broken down to glucose, which is then disintegrated in the process of respiration to release energy, which is used by the living cells to carry out all activities like biosynthesis, like transportation of mineral nutrients, transportation of water, other products for movement, for germination, even for photosynthesis. Respiration usually occurs in the mitochondrion of living cells. However, the process of respiration usually starts in the cytosol of the cell. For today's class, we will be looking at two pathways in the process of respiration. These are the glycolytic pathway and the pentose phosphate pathway. For respiration, we break down the process, the whole series of enzymatic breakdown into three major groups. These are glycolysis, citric acid cycle, and oxidative phosphorylation. Glycolysis is the part that takes place in the cytosol of the cell. This glycolytic pathway can also be called embedded Meyerhoff Pennes pathway. Embeden, Meyerhoff, and Pennes were the three scientists that discovered this pathway in plants. So the glycolytic pathway or glycolysis is the stepwise breakdown of glucose into pyruvic acid. From the word glycolysis, it means glyco is sugar, lysis is splitting. So it simply means the splitting of sugar, which occurs in the cytosol of all living cells. The process does not require oxygen. So be it anaerobic or aerobic organism, the process of glycolysis does not require oxygen. So it is carried out by a group of soluble enzymes located in both the cytosol and the plasmid of the cell. Sucrose, which is usually the storage product of photosynthesis in most plants, or the translocated product of photosynthesis, is a disaccharide which is made up of two monosaccharides of glucose and fructose. So sucrose is first broken down or divided into its monosaccharide or sucrose and fructose for easy entry into the glycolytic pathway. The glycolytic pathway, as we have said, is the first stage of respiration, and this occurs in the cytosol. The glycolytic pathway is divided into 10 steps, or involved 10 steps. The first 4 steps is energy use up step of glycolytic pathway. Now, this glucose coming from the breaking down of sucrose into glucose and fructose. This sucrose goes into the glycolytic pathway, and it is first of all phosphorylated, that is a group of phosphate is added to the glucose in the presence of the enzyme known as hexokinase glucokinase. So the glucose is phosphorylated into glucose 6-phosphate. This glucose 6-phosphate is converted into fructose 6-phosphate, which is an isomer of glucose 6-phosphate. An isomer is simply two compounds that have the same chemical formula but different structural composition. So in the presence of phosphohexose isomerase, glucose 6-phosphate is isomerized into fructose 6-phosphate. Now, again, phosphorylation occurs. That is, fructose 6-phosphate is phosphorylated in the presence of phosphofructokinase to give us a product known as fructose 1,6-diphosphate. This fructose 1,6-diphosphate is broken down into two triose phosphate, that is two group of carbon 3 phosphate, which are glycidiide 3 phosphate and dihydroxyacetone phosphate. The dihydroxyacetone phosphate does not go directly into the next stage. It is converted back into glycidiide 3 phosphate. So for the next stage, which is the energy producing phase of glycolysis, you'll be having two molecules of glycidiide 3-phosphate going into that process. Remember, in the first stage here, we have used two molecules of ATP. So going into the energy producing phase of glycolysis, you have the glycidiide 3-phosphate, that is two molecules of glycidiide 3-phosphate being converted in the presence of glycidiide 3-phosphate hydrogenase into 1,3-diphosphoglycerate. In that process, two molecules of NADH are produced. Now, the 1,3-diphosphoglycerate is dephosphorylated to produce 3-phosphoglycerate. That is, a molecule or two molecules of ATP are produced from the two glycidiide 3-phosphate to give us 3-phosphoglycerate. In the presence of another enzyme known as phosphoglycerate mutase, the 3-phosphoglycerate is converted into 2-phosphoglycerate. These 2-phosphoglycerate then loses a molecule of water. That is, dehydration occurs in the 2-phosphoglycerate to give us phosphoenol pyruvate. The phosphoenol pyruvate is dephosphorylated. That is, a group of phosphoride, a group of phosphate is removed from the phosphoglycerate to produce pyruvate. So in this second stage, you have 4 molecules of ATP being produced and 2 molecules of NADPH being produced. So at the end of the glycolytic pathway, you'll be having 1 molecule of glucose producing 2 molecules of ATP. Remember, the first stage, we use 2 molecules of ATP. In the second stage, we produce 4 molecules. So the net balance will be 2 molecules of ATP, 2 molecules of NADPH, and 2 molecules of pyruvate are generated at the end of glycolysis. Now, in the presence of oxygen in such a cell, the pyruvate goes into the mitochondria, where it is completely broken down to release the energy stored in it, because this pyruvate has loads and lots of energy in it. It is carrying the bulk of the energy that is needed in the cells So if such a cell has oxygen, that is, oxygen is present in such a cell, the pyruvate goes into the mitochondria to be completely broken down into um, energy, carbon dioxide, and water. But if the cell lacks oxygen, the pyruvate remains in the cytosol where where fermentation occurs to give us acetyl or to give ethanol, which are products of alcohol, and the energy remains stored in this alcohol. So such a cell will not really have the energy. In the process of glycolysis, the direct synthesis of ATP is known as substrate phosphorylation. That is, in the process of carrying out glycolysis, you have substrate phosphorylation taking place. That is the production of ATP. So that is for glycolysis. The second pathway we want to look at is the pentose phosphate pathway, which kind of simultaneously takes place along the glycolytic pathway. This is a branch, is a branch off from the glucose 6-phosphate. That is, when the glucose 6-phosphate is formed, a part of it, enzymes and electrons are moved out. So this pathway runs alongside the glycolytic pathway, and it is carried out by soluble enzymes present in the cytosol and in the plastids, just